All right, but we have a lot of ground to cover, so I think maybe we will go ahead and get started. Because as I say, we have a lot of things to talk about, and really want to say up front, want to make sure that this is a conversation. And I'll just say from a logistics point of view, throughout the program, feel free to put your questions, comments in the chat feature. I'm going to be keeping track of that as best I can. You also, if you have trouble with the Zoom interface, which I'm sure is too familiar to too many of you now, months in, to this new abnormal, you can certainly always email avac@avac.org with questions, and then we will also be able to raise hands in the Zoom interface. Again, I'm Mitchell Warren, the director of AVAC, and delighted to welcome all of you, and particularly to welcome Barney Graham to this webinar that is remarkably timely about COVID vaccine development. And there are obviously many people working in vaccine development right now for COVID. Many of you on the line as researchers, as advocates engaged in this process. And I'm delighted because I can't think of really anybody better suited to give us a state of the field and an update, particularly on one of the vaccines in phase 3 trials right now than Barney Graham. As you can see from the slide, Barney is the Deputy Director of the Vaccine Research Center. For those of you who don't know, and we talk about this very often every May in Vaccine Awareness Day, May 18th is recognized as HIV Vaccine Awareness Day because then President Bill Clinton made a really remarkable speech at Morgan State University talking about the 21st century being a century of science, a century of vaccine development, particularly for HIV. And one of the great outputs of that speech of 1997 was the establishment of the Dale and Betty Bumpers Vaccine Research Center at the NIH. For those of us, and many of you on the line, I see your names, have worked with AVAC, with Barney, with the VRC around HIV vaccines for many years. And that's certainly where the VRC began. But the VRC has worked over its now more than 2 decades on more than just HIV, around other vaccines and development that you'll hear Barney describe, certainly around now COVID-19 vaccine development. Also, as many of you will know, they've really pioneered work in neutralizing antibodies, and we all are looking forward to initial results from the VRC's own antibody in 2 large trials, the results of which are coming out later this year, the AMP studies around the VRC-01 antibody. So you really get a sense of the VRC's leadership across product development, across disease areas, and we are delighted to have Barney here today. I do want to also say, just really as a bit of a frame, and just to remind people, just yesterday we at AVAC released 2 documents that we hope are helpful to everyone on the line. They're available at avac.org. One is a bit of a cheat sheet. It looks at the pipeline of vaccine candidates for COVID-19, those that are in later stage human efficacy trials, as well as those candidates that have received significant support from the US government as well as from CEPI, and working in the larger COVAX, COVID vaccine accelerator through the World Health Organization and Gavi and CEPI. So that cheat sheet is available online and walks you through some of the vaccine constructs, where the funding is coming from and at what stages they're in. We also presented a document that we're really excited about, talking about the risks and benefits of expedited COVID-19 vaccine research. For any of us who've worked in HIV vaccine research, it is really kind of awe-inspiring to see the urgency and the speed with which investments have been made in COVID vaccine development. And I think you'll hear Barney describe some of that work. That speed is necessary, the urgency is necessary, but of course never at the expense of scientific process, of safety, of ethics, of community engagement, of thorough independent regulatory review. And this advocate's guide to the risks and benefits of expedited research, we hope is helpful to all of you as advocates. And again, those are available. And thanks, Levi, for putting in the link to both of those documents that are available online now. Also, if they're always, with traditional AVAC documents, they're always works in progress. Please do feel free to send us comments, additional questions that may come from that. And again, happy to take any of those questions later in this call. Again, we are muted for everybody right now except for Barney, so that he can present. We are recording this, so it's available to those who can't join this call. But without further ado, I'm delighted to welcome Barney Graham from the Vaccine Research Center at the NIH to provide a really critical update around COVID vaccine development.
Thank you, Mitchell, and it's a pleasure to be with you and with all of our friends from HIV vaccine development over the years. And I'm going to try to connect HIV and HIV vaccine work with what's happening now in COVID, because almost everything that's happening is related to our original efforts in HIV, and including the rapid approach. And even though we don't have an HIV vaccine yet, when I joined the VRC back in 2000, we were still having 14,000 new infections every day. And the agreement among all the investigators who went to the VRC was that we would not waste any time, because every day was another 14,000 people. And so we made a commitment to not let documents sit on our desk overnight and not delay. So this way of working, even the way of working now for COVID, is really related to everything that has been set up for HIV. As Mitchell said, this our Building 40, the HIV vaccine, the VRC, the Vaccine Research Center, was proposed in 1997 by Bill Clinton at Morgan State, and the center was established to focus on HIV vaccine development. But in the meantime, with all the technologies that have been developed because of work on HIV, we've been able to address other long-standing problems and unmet needs like influenza, Ebola, respiratory syncytial virus, and also a number of emerging infectious diseases shown here, using the technologies from HIV. And that's possible for us because we're privileged to not only have a basic research building, but have process development capacity with about 100 engineers north of us in Gaithersburg and another 100 or so people in a pilot plant in Frederick that can make GMP material. We have our own self-standing clinic in the clinical center, and Margaret McCluskey's on the line. She helped me start this many years ago. And a separate laboratory to do GLP analysis of the clinical samples. We have made over the years a number of different ways of delivering vaccines, including nucleic acid vectors, virus-like particles, for instance, chikungunya, now enterovirus, number of protein subunit approaches that we'll talk about, and then, as Mitchell said, monoclonal antibodies. We've sent these products or candidate products all over the world, and it's done with this small group of investigators, PIs, and program heads, and led by John Mascola here in the middle. So over this last decade, we've had so many new emerging infections that it just felt like an onslaught to us, because we responded to many of them, starting with chikungunya and MERS back in 2012. And then Ebola and Zika and more Ebola, and now this new coronavirus. And so we've been thinking really for several years about how can we do pandemic preparedness better, or more proactively, more prospectively. There's three major concepts that we've thought about and used, and one is the platform approach, which means platform manufacturing that can be plug and play and avoid a lot of the repetitive need to establish methods of manufacturing and purification. This allows you to use the same platform regardless of immunogen. Priority pathogen approach is something that WHO and CEPI, the Coalition for Epidemic Preparedness and Innovation have used selecting either 8 or 3 major pathogens of concern. And we've advocated for a more systematic approach to all 25 virus families that are known to infect humans in what we call a prototype pathogen approach. And I'll explain what that means a little more later. We think this is possible, again, because of what HIV vaccine efforts have done. And that is, they've led to a number of new technologies that give us more precision in how to make vaccine antigens, how to deliver them, and how to analyze them. And I'll talk more about structure-based vaccine design. But this ability to do single cell analysis has led to rapid identification of monoclonal antibodies and definition of lineages, and ability to analyze immune responses in more detail, and also new ways of displaying antigens on self-assembling nanoparticles. And that gives us a precision that can be matched with the speed of platform manufacturing and these other technologies that give us abilities to make rapidly make new cell lines and animal models if needed. And I'll talk more about the nucleic acid approach for manufacturing. So for me, this started really back in 2010 when we started a program with Jason McClellan and Peter Kwong's lab at the time at the VRC to understand the structures involved in RSV, respiratory syncytial virus immunity. And eventually, by 2013, we were able to solve or capture this structure of the pre-fusion form conformation of RSV-F. The F glycoprotein is a class 1 fusion protein analogous to the spike protein on coronavirus or to GP160 on HIV. And what we found was in the process of defining this structure, we identified new antigenic sites, especially at the apex of this prefusion, that are more neutralization sensitive than previously known sites. And it's because the previously known sites are also retained on this post-fusion form of F. This is the same 574 amino acids. It's now been rearranged in the post-fusion event because this molecule is what mediates viral entry and mediates membrane fusion. And so when this rearrangement occurs, it can even happen spontaneously on the viral surface, you lose this epitope that we call site 0. And this protein, which has been used repeatedly over the last several decades trying to make a vaccine for RSV, has never been able to boost responses more than 2 to 3-fold on average. But after we had this structure and then stabilized it with the C-terminal trimerization domain and some internal disulfide and cavity filling mutations and maintained the original native structure, we found that you could now boost responses by up to 16-fold, neutralizing activity up to 16-fold, which turns it into something that now can be a vaccine antigen. So just trying to understand the atomic structure revealed this new target of vulnerability. We now have clinical proof of concept for structure-based vaccine design, and the idea that preserving neutralization sensitive epitopes at the apex of a class 1 fusion protein may turn it into a good vaccine antigen. And so we've now applied this to coronavirus and also some other paramyxoviruses. So as we found this, it was at the same time MERS had come into the Middle East and was another pandemic threat. And we started trying to solve the structure of the spike protein that had never been seen before on coronaviruses. And so Jason McClellan, it was now at Dartmouth, continued to be my collaborator on this kind of thing. And this protein is very large. It's almost 3 times bigger than the RSV-F and twice as big as GP160. And X-ray crystallography didn't work. And so Jason involved Andrew Ward at Scripps to do cryo-electron microscopy. We still couldn't get the MERS or the SARS, but after serendipitously starting a project on HKU1, an endemic beta coronavirus, this was inherently more stable, and Andrew's group was able to capture the pre-fusion structure of the spike, shown here. And then Jason's group and us were focused on how to stabilize this. They found 2 proline mutations at the top of the central helix, largely stabilized this protein and kept it pre-fusion. It turned out that these 2 prolines could also be translated into other coronaviruses, and including all the beta coronaviruses, and put into an analogous position at the top of this central helix, could stabilize MERS and SARS. So now we can have the structure of MERS and SARS spikes. We have the apical epitopes preserved. And equally important, especially for gene-based approaches, not only preserve the important epitopes, but it increased expression levels. So for instance, MERS was expressed in the 2P stabilized format 50-fold higher than the wild type sequence. And so since we've had 2 beta coronaviruses within the last 10 years, and it's clear that coronaviruses are going to continue to emerge in the human population, shown here for the first 4 endemic coronaviruses, originally from an animal reservoir, likely going through an intermediate host, that we're going to continue to have these problems. So the 2 alpha coronaviruses endemic, shown here, and the 2 beta coronaviruses that are endemic, shown here, are now joined by 3 other beta coronavirus, SARS-1, MERS, and now SARS-2. And so we wanted to combine the structure-based approach, the precision approach, with a rapid manufacturing platform. And because of our work with Moderna and Zika during 2016, we adopted their mRNA strategy, which we knew could be synthesized quickly, and based on our work with them in 2016 was a very potent method of antigen delivery. And it works by encapsulating the RNA into this lipid nanoparticle that delivers, not only stabilizes, but delivers the RNA into the cytoplasm and can be translated then into a protein that can then look very much like a protein that would be placed onto the viral membrane and elicit antibodies to conformationally correct epitopes. And one of the reasons this works is because of the chemistry and manufacturing approach to the mRNA. Some of this was originally learned by Drew Weissman at University of Pennsylvania, in that if you remove the wild type uridine and make a 1-methyl or 5-methyl modified uridine nucleoside to include in your mRNA, that it reduces the overall triggering of these internal detectors, these pathogen-associated molecular patterns. And so making a modified uridine reduces this signaling effect that creates interferon responses. And then in the manufacturing processes, removing any of the double-stranded RNA intermediates that would be generated prevents this other set of responses through TLR3 and RIG-I. And so if you make the RNA and deliver it this way, it's more of a stealth, under the radar type of delivery, and it allows the protein to be expressed and displayed longer on the transfected cells, and we think that makes it more immunogenic. So the first project was with MERS, coronavirus spike, and we made the MERS stabilized spike with mRNA from Moderna, and have tested that in mice, and challenge mice then that had the human DPP4 receptor, and showed that 2 doses of either 0.1 or 1 microgram could prevent infection of lung and prevention of the disease that can be seen in this model, and completely prevented weight loss at these two relatively low dose levels. And even at the breakthrough dose of 0.01 microgram, the partial immunity resulted in partial protection. It didn't cause the enhanced disease that some of you may have heard about as some concern with coronavirus. So we now have the proline mutations that stabilize spike. We know it makes it more immunogenic. We think this could be a general solution for beta coronaviruses. And now we have a rapid manufacturing platform in case these things ever happen again. And we had already set up demonstration projects for both Nipah and coronavirus and MERS with Moderna, and we're planning to take the Nipah forward into a phase 1 trial when this new outbreak was announced from China. And by January 7th, we had heard that it was likely to be a beta coronavirus. And so we switched from our Nipah project to a coronavirus project, and we decided to run this drill on rapid platform manufacturing for new pandemic threats, and thinking of the MERS as a prototype for the virus family of coronaviruses. And so the sequences were released the night of January 10th. We designed constructs, ordered reagents on the 11th, and by a few days later had agreed with Moderna on which sequence to take forward. And Stefan Boncel, to his credit, made a decision at risk to manufacture. And within 41 days, we had GMP product that could be used to start a clinical trial. And at the same time, DMID helped us to file IND and set up a phase 1 trial within their network system. So it was a it was intramural, extramural, and industry all cooperating to rapidly move this into a phase 1 trial. And so within 65 days, a phase 1 trial was able to start. And then by May 29th, phase 2 started, and by 27th, the phase 3 started. And people have been nervous or worried about how fast this has gone, but I can tell you, at least from a scientific and data perspective, we haven't skipped any steps. And before we started phase 1, we knew this product was immunogenic. Before we started phase 2, we knew it could protect animals. And before we started phase 3, it had satisfied the safety criteria that we had imposed on it, not just for local reactogenicity, but for the potential for any kind of vaccine-enhanced illness. So none of the data steps have been skipped here. I think the things that has been put at risk is the expenditure of money and a lot of time. So time and money were put at risk, but I don't really think, at least up to this point, that any kind of safety has been put at risk. The first thing that we did with Jason again, McClellan again, who's now at UT Austin, was to make proteins so that we could get the structure of this spike on the new virus. And that was done within just a few weeks. So shown here is the spike protein of the coronavirus, the new novel coronavirus. And this is the receptor binding domain sticking up. This is also a trimeric protein, but it's got a different structure than GP160 that you may be used to, or even F of RSV. And these receptor binding domains flip up one at a time in order to engage the receptor ACE2, because the ACE2 binding site is actually under this RBD, and if it doesn't flip up, it cannot be engaged. And since we didn't have reagents for this, antibody reagents, it created a lot of motivation and confidence that it was in the right shape. So we had a spike protein in the right conformation and shape that allowed us to create diagnostic assays as we did with the CDC. And use it as a B-cell probe, as we did with Abcelera and Lilly, who now have that first antibody into clinical trials, even into phase 3 trials. Or we could apply it to vaccines, as we did for mRNA and as others have done for these other approaches, either nucleic acid or vectors or protein-based approaches. And so on the WHO website, it lists, this was last week's, it's probably changed since then, but there's at least 28 vaccine candidates for coronavirus in clinical evaluation. They are either nucleic acid, whole inactivated virus, vector-based approaches, or recombinant subunit proteins. And there's another at least 139 candidate approaches being developed by other labs around the world using these other kinds of platforms, nanoparticle display, other vectors, live chimeric viruses, etc. And in some presentations, people are listing upwards of 223 vaccine approaches being taken by various labs around the world. But I'm going to focus in on the ones that are in the most advanced stage in clinical development. And I'm going to give you the data that was the basis for us taking the Moderna product forward into phase 2 and phase 3 trials. First, with just a couple of slides from mice, we used a 0-3 regimen of mRNA delivery and looked at serology, did 4 weeks after the second dose, or 13 weeks after the second dose, or 7 weeks after a single dose to look at protection. This was done with Ralph Barrick at UNC, who, while we were measuring the response, was developing a mouse-adapted virus that has 2 amino acids changed in the receptor binding domain to allow it to bind the mouse ACE2, and now has been passaged and adapted so that it can actually make mice sick. And we think this is a really good model. It can infect wild-type mice and allows you to do a lot of detailed immunology. So 2 doses induced decent responses by ELISA and also neutralizing activity by pseudovirus neutralization up in the 1,000 range, and even a single dose of 10 micrograms could induce neutralizing activity up to 1,000. At those levels, 1 microgram completely protected lung from virus challenge in both the lung and the nose, and the 10 microgram and 1 microgram, even at a single dose at 7 weeks, were able to protect the lung. And at 13 weeks, you could also continue to show protection of both lung and nose. And then looking at the breakthrough infections where there was only a 2 log reduction or maybe a half log reduction in nose with these breakthrough doses, you could show that there was no evidence of serious immunopathology, and that in the PBS control group, you could see acute lung injury effects with these neutrophilic nodules and increased alveolar septa and infiltrates, interstitial infiltrates by neutrophils and lymphocytes. But these effects didn't happen in even the breakthrough infected animals, and certainly didn't have any sign of enhancement of pathology or altered cell content in the pathology. We also have looked at aged mice and asked whether these types of products would work in aged mice, comparing 2 doses of mRNA to a whole inactivated virus that was derived from the old SARS-1 and inactivated in a way that disrupted the spike protein and made it and ruined some of the epitopes, because formalin is hard on those antigens. And so you could show that in aged mice, if anything, the antibody responses were higher than in the younger mice. And even the neutralizing activity was now up well over 1,000, approaching 10,000 levels. And even though it was SARS-1, there was some cross-reactive neutralizing activity against SARS-2 in this whole inactivated virus immunized mice. And you could show that at the 1 microgram level, you prevented illness. This is now a virus that's been passaged and adapted and can make mice sick. So the 1 microgram prevented virus replication in lung and nose, and also prevented weight loss. The 0.1 dose that reduced virus by 2 logs had some modest but controlled weight loss, and then the whole inactivated virus that didn't protect at all, or maybe even increased replication, caused illness similar to what was seen in the PBS control. And though that virus, the whole inactivated virus that was formulated in alum, also did this thing that has happened before with respiratory syncytial virus in the 60s, when whole inactivated virus or vaccines that poorly induced neutralizing activity also created a Th2 dominant response post-challenge. And so you can see the whole inactivated virus is now primed and induced these Th2 responses, which are well above what was seen even in the PBS controls. So both E-attacks and chemokines and other type 2 associated cytokines were induced by this whole inactivated virus prep, but not by the mRNA at all. In fact, the 1 microgram dose that protected also prevented all these inflammatory cytokines that came up either in the PBS control or in the whole inactivated virus control. So preventing infection with the mRNA at a protective dose also reduced all these inflammatory cytokines. We looked at this next in monkeys, non-human primate rhesus macaques. And here we have groups that had PBS control or a 10 or 100 microgram dose of the mRNA. And looking at binding antibody, all the animals responded to the first dose in terms of the lysa binding activity, and after the second dose, boosted. You can see these responses at either a 10 or 100 microgram dose level compared to convalescent, a panel of convalescent human sera. And if anything, even the 10 microgram dose level is in the upper tier, or in the upper half of convalescent responses, and 100 microgram is well above convalescent responses. Focused in on just the RBD binding domain, this is whole spike trimer, RBD bind specific subdomain, and this is the N-terminal domain, the knob on the corner of the top of that spike is the N-terminal domain, and even that response is above or in the upper tier of convalescent sera. Looking at neutralizing activity or ACE2 binding inhibition, this is preventing ACE2 from binding the RBD in an assay that's based on MSD technology, and you can see that this is again well above, or at least in the upper tier of convalescent sera. And using the live virus neutralization from Ralph Barrick's lab, nanoluciferase reporter virus, again you can see neutralizing activity well above convalescent sera. T-cell responses also showed a Th1 dominant response, and so these are blood PBMCs stimulated with peptide pools from spike, and almost all the response is CD4 Th1 bias. There's no Th2 associated cytokines or T-cell responses, and you were able to see T-cell responses in all the animals, and those were the CD4 T cells were largely making IL-21, which is associated with T follicular helper cells that are important for activating and expanding germinal center B cells to become better antibody producing plasma cells. And this is something else Drew Weissman has shown in the past, that this approach where you use a stealth mRNA that doesn't induce a lot of these innate responses is particularly good at inducing T follicular helper CD4 T cells. So in challenge of the macaques, I'm showing you subgenomic RNA, which means RNA that is generated during replication of virus. This is not representing the whole virus genome PCR that would be partly from the inoculum, but only looking at replicating virus. And what you see in the controls is that in the lung, you have on day 2, over 5 logs of virus. And then it is relatively rapidly cleared by day 7 and drops a lot by day 4. But in the either 10 microgram or 100 microgram dose groups, you have largely prevented virus replication in the lung, or represented by the BAL. And even in the upper airway, shown here in the control, this is nasal swab replicating virus. 10 micrograms may have had some early clearance, but the 100 microgram dose group had very little, if any, subgenomic virus, replicating virus in the upper airway. And so at the time, this was the first demonstration of protection of both lung and upper airway, but now there's been other examples of that from Dan Baruch and other groups using other types of approaches. And then for pathology, you see there is some level of pathology and interstitial infiltrates in the PBS control groups, and you can demonstrate virus either by RNA hybridization or by antigen immunostains. No virus was detected in any of the control group, in any of the vaccinated groups in the lung. There were just very minimal peribronchiolar infiltration of lymphocytes in the low-dose vaccine group, which are considered protective type responses. The thing that was associated or correlated best with protection was the antibody responses to the RNA in the upper airway. And so you see, especially with live virus neutralization, that you got nice correlation curves with antibody, higher antibody, lower RNA in the upper airway. So the human data, all these things were proceeding in parallel, and I'll show you data from this younger age group in adults at 2,500 and 250 microgram dose levels. There's now a paper that's been submitted demonstrating the immunogenicity and safety in these older age groups as well. And the phase 3 trial will be based on the 100 microgram dose group and include people over 18 years of age up into the very older age groups. This was given on a 0-4 week schedule. And for safety, you may have heard a lot about this. So these are local and systemic reactogenicity profiles, and gray is mild, meaning you can feel it, and blue is moderate, meaning that it bothers you somewhat, and the red, the severe, would be something that required or led you to take ibuprofen or Tylenol for the symptoms. So most of the blue and red marks you see are after the second dose. That's one finding. Most of the red responses are in the 250 microgram dose level, which is the third bar in each of these boxes. And so the second dose and the high dose level is the one that was considered too reactogenic, causing things like just soreness and pain at the injection site and some fever and arthralgia as systemic symptoms. But even at the 100 microgram dose level, you got very few, if any, severe reactions, and there was no serious adverse effects, meaning that somebody had to seek medical attention or go into emergency rooms, things like that. at 100 micrograms or lower. So this was considered to be a well-tolerated vaccination. And in these younger adults, the 18 to 55-year-old dose group, this is now looking at full-length spike trimer, or RBD, binding antibody. At all dose levels, you could see responses after the first dose in all subjects, and all subjects boosted after the second dose at day 29. And the responses you can see are sustained reasonably well through day 57, which is just 1 month after the second dose. And that the 100 microgram dose group and the 250 have very similar responses. And so going with the 100 microgram dose was an easy decision because of the reactogenicity and the immunogenicity both suggested that it was good enough. So this is the receptor binding domain specific antibody response, again showing responses that were relatively sustained for a month after the second dose. And all of these are higher in the upper tier of things that would be seen in convalescent human sera panel, shown here on the right. This was also true in the pseudovirus or live virus PRNT neutralization assays. And after the 2 doses of 100 micrograms, you saw nice responses that were sustained at least through day 57 that were in the upper tier of what you see in convalescent sera panels, and same is true even to a larger extent in the live virus neutralization, which unlike most of the pseudoneuts, this live virus neutralization is more sensitive or gives you higher levels than the pseudovirus neut. And these levels are about 3 times higher than average convalescent titers, and these are about 5 times higher than average GMT of convalescent serum panels. The T cell response was also consistent with what we saw in the monkeys, in that people made decent CD4 T cell responses, and they were Th1 biased with very little Th2, if any, cytokine production. The CD8 T cells were detectable, but they were at very, very low levels. So we have evidence of immunogenicity now in mice, monkeys, and humans. We showed protection in mice and monkeys, and we have shown safety data, not only from neutralizing activity, but from an appropriate Th1 biased T cell response. And that's what's allowed us to enter into phase 3 studies with some level of confidence. And we're not the only group who's done this. Moderna is the group I'm talking about, and our phase 3 study started around July 27th. And we've now enrolled a little over 14,000 people, so almost halfway to the 30,000 subjects in the study. And then we'll start collecting endpoints 2 weeks after the second dose in all these subjects. Pfizer also started their phase 3 trial on July 27th, although we started in the morning and they started in the afternoon, if you want to know. And they are also using an mRNA, and it turns out that it's also expressing the same construct, the spike protein with the 2 proline mutation. Other groups that are soon coming into phase 3 include AstraZeneca, which makes a chimp ad vector, a single round replication defective adenovirus vector that's a type E ad vector. And they should be starting phase 3 trials very soon. They do not use this S2P mutation. But the next group, Novavax, who makes a subunit protein, is using the S2P stabilized version and made in baculovirus. Janssen makes an Ad26 adenovirus vector, a type D ad vector, and also expresses the S2P construct. Sanofi, collaborating with GSK for their AS03 adjuvant, is also making a baculovirus-produced S2P stabilized spike construct as a subunit protein. I thought I would tell you briefly before I stop how all this is being organized in a new way, because this is another way HIV has really impacted the ability to respond to coronavirus, this new coronavirus. And a new structure was established within the government called Operation Warp Speed, OWS. And I think you've heard Monsef Slaoui was appointed to lead this, and it is helping to facilitate diagnostics, largely led through the CDC, therapeutics, largely led through FDA, and vaccines led by DOD and NIAID. Within the vaccine program, there is a group led by John Mascola on vaccine development, another one led by BARDA on supply and manufacturing, and another by CDC on delivery and administration. And within the vaccine development group, there are other subcommittees on preclinical immune assay development and managing the phase 3 clinical trials. And for the phase 3 trials, these are all groups that have been sponsored by BARDA, using BARDA funding. And so in these phase 3 trials, it's been a new consortium of networks coming mostly out of the HIV vaccine trials network, the HIV prevention trials network. The ACTG is also involved in some of the therapeutic studies. And this IDCRC, which is a derivative of the old VTEU system, and then some DoD sites as well, make up a new coronavirus prevention network. And they have sites scattered, almost 60 sites scattered throughout the United States. And you can see now in a map divided by county how many types of how much coverage there is, because it's not only the co-VPN sites, but also contract sites from PPD working with Moderna. So this new way of doing phase 3 is a hybrid of a contract research organization like PPD working with Moderna and the government network sites, which has posed some logistical and philosophical problems along the way, but they're getting sorted out, and in an unprecedented way are going to lead to a number of phase 3 trials within a year from this virus emerging, which is remarkable. There is a coronaviruspreventionnetwork.org website where you can learn more about these different products coming through, and also register as a potential volunteer for these studies just by going through a short questionnaire. And then this data from the counties is overlaid with information on incidence rates, location of places like meatpacking industries, other places where there may be hotspots, and so there's something called predictive analytics that is helping to guide the enrollment rates at different sites around the country. So in summary, in my way of thinking, this all really started because of fundamental, basic, and translational research that had been done in the past. Combining precision vaccinology and new technologies driven largely by HIV vaccine efforts. This concept of prototype pathogen preparedness, where you can take one viral family member, understand everything about it, know what to do if another related virus comes, and have at least a sense of better preparedness. And then having in place ahead of time these public-private partnerships, which allowed rapid manufacturing to take place without any additional experimentation. And this mRNA-1273 We know it can protect mice and non-human primates in both upper and lower airway. We know it's immunogenic in older age groups, and the phase 3 trial, as I mentioned, was started on July 27th. And I just want to thank all the investigators at the BRC who all have contributed to this. We have really stepped out of our routines, and every single person up here has contributed to coronavirus. Jason McClellan, who's our long-term collaborator on structure especially, and within my lab, Kazmikia Corbett, hiding here, has led our small coronavirus team before this outbreak started, and is assisted by Caitlin Morabito, who is an important project manager for this project. And then, of course, Moderna, Mark Dennison at Vanderbilt doing neutralizing assays on live virus, and Ralph Barrick, who's established much of the virology technology that's been used to make new viruses and animal models. I will stop there, Mitchell, and take any questions if we have time.
Great, Barney, thank you so much. And we will make time. A lot of great questions have come in already, both in the chat and the Q&A, and then some emails separately as well. We're going to try to get to as many as we can. Barney, you can stay a little past the top of the hour, I hope?
Yeah, I can stay until we're finished.
Great. So I'm going to do my best to navigate through these in various orders and bundle a few together. A number of people, not surprisingly, asked, when will there be phase 3 results? So maybe just a quick sense. So you talked about the 30,000 person trial, enrollment moving quite swiftly. When might you expect to see results from the phase 3?
Well, it depends on, we have an interim analysis planned when there's 53 clinical endpoints that are proven to be coronavirus infection. And we think we'll have a good chance at finding an answer if the vaccine's at least 50% protective or 60% protective by after we have 151 clinical endpoints. So it really depends on how these endpoints accrue, and we're hoping that the predictive analytics and shifting recruitment into various places that we anticipate will be hotspots will allow us to collect endpoints. But you know, we won't be to 43 days after the enrollment of the last subject until, you know, around early November. And so if we get all these endpoints out of the first 10,000, it could happen earlier. If it takes all 30,000 enrollments and waiting until the winter epidemic, which we're all afraid might be bad this winter, coronaviruses, at least in North America, usually are peak between December and the end of May. So we don't know. We don't know when these cases will accumulate. So this is an endpoint-driven trial. We have to wait for the endpoints.
That's super helpful. I think obviously, at least for those in the US, a lot of concerns about the politicalization of issues. And so, you know, being grounded in the science of what data will be available and having a sense of that process is really important for advocates, particularly to pay attention to. There was a question about endpoints actually, and about what is the endpoint. And I think it might be helpful just to have you describe, Barney, what you expect this vaccine to do. A lot of conversations about you know, the severity of disease compared to preventing infection at all. And we had some of these same conversations back in HIV vaccine days a decade or so ago. So maybe it'd be helpful just to describe what you expect and what you're looking for in the actual trial and as designed for endpoints. Right.
Well, this is a respiratory virus. This is not exactly what we're used to. It's a different biology than what we're thinking about for HIV. And so in respiratory viruses like respiratory syncytial virus or influenza or now coronavirus, the primary goal, the first goal of a vaccine is to protect the lower airway because that prevents the most severe disease. So if you can have a good enough antibody response that can penetrate and transduce or transfer from the serum into the tissues of alveolus and lower airways, you should be able to protect against severe infection and severe disease in that way. If you can get a really good antibody response in which the transudate can penetrate the upper airway where the gradient is much higher, then you may be able to also reduce nasal infection and upper airway infection and have an impact on transmission. And so, as you saw, we, in monkeys at least, which is a semi-permissive model, we were able to impact both the lower airway and the upper airway as we were in mice. Can we achieve that level of efficacy in humans is an open question, but the first goal is to protect the lower airway. Secondly, we want to protect and reduce transmission by decreasing virus in the upper airway. Great. And you know, most of our respiratory virus vaccines, the main one we have licensed is influenza. It's not a very precisely made vaccine. It's whole inactivated split product without much regard to the content or the conformation of anything. And we know that at best, it's only 60% effective at preventing disease, influenza-based disease. And in some unmatched years, it's less than 10% effective. And so we're hoping that this vaccine, you know, I'm hoping it's going to be more in the 70 or 80% category, you know, maybe in some people up to 90%, but we don't really know that yet. And we have to wait and see what the dynamics are. So our main goal is to prevent infection and severe disease. So the endpoints are clinical, like most vaccines. If someone has a symptomatic infection, symptoms of a respiratory infection, and they are PCR positive, that will count as an endpoint. Those people will be followed to see how severe the disease becomes, because typically in respiratory virus vaccines, it's easier to show efficacy against severe disease. And as you get milder and milder down to just simple infection without symptoms, it becomes harder and harder to show efficacy. You may need very, very large studies to show efficacy against just infection of the upper airway, for instance. And so I'm hoping that we get really high efficacy against severe disease and pretty good efficacy against any symptomatic disease. And we'll just have to see.
And on one of those last points, no matter what you see in an early interim analysis, just to be clear, the follow-up will continue, correct? To see, because there's a question of durability, you know, could this be more like a flu vaccine where you might need it every year? So what will happen, say, post-November, December, January?
We're starting some monkey studies to look at durability and, you know, memory that would be at least a year. In the clinical trials, the companies have been asked to follow people through 2 seasons, which means 2 winter seasons. So it would be a 2-year follow-up. Okay. But if the interim analysis, it gets a little complicated what you'll be able to learn, because if the interim analysis shows, let's say it shows it's 80% effective, then there's a bunch of placebo recipients in that trial who really would like to get the vaccine. And if they get the vaccine, we won't get 2 years of follow-up on the vaccine unless we can figure out a way to analyze it by staggered and, you know, staggered immunization. And so if the vaccine is really effective and we start immunizing everyone, we'll have less long-term data until the long-term gets here, which means we won't really know unless things start failing in the winter of 21, 22. And so I think that durability is going to have to come with time, and it's just we're just going to have to wait.
Yeah. Speaking of time, age matters for many vaccines, and you talked about the phase 3 looking at 18 and above, and you said you described very old, and I'm sure we all have a different definition of very old. Obviously, older individuals are going to be in greatest need of this vaccine. What's your sense of differential immune responses by age, and might you know, when you look at the recruitment, if you see great efficacy at a lower age range, you know, what's the concern that about perhaps older people having a different immune response, and how do you deal with that?
Well, there's going to be a big emphasis here on at-risk groups, which includes the elderly. And so most of the studies in phase 3 are going to include people all the way up into their mid-80s. And hopefully we'll have enough numbers there, and there'll be enough severe disease to really show that it works in these different categories of people. And but you know, the emphasis is on the elderly in part. And the first clinical trial that published data from the elderly just came out from Pfizer. It's posted. It's not peer-reviewed yet, but the Pfizer vaccine that uses the full-length spike, the stabilized full-length spike, similar to what we're using, shows that you might have some drop-off in the older age groups, but it's not profound. And so, and we have data now that I mentioned was being prepared for publication, where it looks like we're maintaining pretty good responses in the older age groups. So, and I showed you data from old mice today, and those old mice made maybe even better responses than the younger mice. So I think that we'll have at least some vaccines that can have decent immunogenicity in older age groups. So we should be able to get an answer in those groups, we hope, in the phase 3.
And Barney, you know, talking about the Pfizer data, you know, for the mRNA platform or concept, it's relatively new. There's no current licensed vaccine, correct, using mRNA? And yet here we have these 2 phase 3 trials using it. And so you did a great job of explaining the mRNA platform generally, but are all mRNAs the same? I'm guessing the answer is no, because you wouldn't both be developing these vaccines. There was a couple of questions about the dosing. I think the Pfizer dose is much smaller than the Moderna dose.
Right.
And if you could maybe describe what's different for the layperson.
Well, there's a number of different ways to deliver an mRNA vaccine. So there is something called an amplicon that comes out of alpha viruses, in which the message that comes off of RNA can be self-amplified. So a number of companies have this technology, including BioNTech, which is one of their vaccines in phase 1, was a self-amplifying mRNA, where the mRNA that you put in can make a massive number of transcripts that increases the amount of potential protein that can be made, but it also really induces all these innate receptors that can create innate responses that might shut it down pretty quickly. So the philosophy there is you make a lot of protein, even though it may not be made for very long, you're making a lot of protein. Another approach is just to use normal nucleosides, unmodified nucleosides. And so those would be inducing some innate response, but maybe not as much as the self-amplifying. And they would make a reasonable amount of protein and would also induce, sort of be self-adjuvanting, and maybe add an adjuvant effect to whatever protein was being produced. And then Pfizer, BioNTech, and Moderna have all made modified nucleoside RNAs, which try to reduce the amount of innate signaling. And then another modification that I tried to briefly mention was some of these companies have more or less double-stranded residue in their vaccines, which could induce more or less TLR3 and RIG-I type of messaging. And so there's probably at least 4 different categories of mRNA that could have different kinds of outcomes. And so it is a little bit nuanced.
Yeah. And that's super helpful. And I do want to let people know we are hoping that one of the developers of the self-amplifying RNA is Robin Shattuck's group at Imperial College in the UK. And we've been talking to Robin about scheduling a call like this just to get a sense of the diversity of the pipeline.
Right. Robin is working on a self-amplifying RNA concept. So yeah, he could talk more about that.
Yeah, no, that's great. I wonder, you know, Barney, when you look at that great slide you had of all the different programs, you know, if you do get an early signal of high efficacy, is there a conversation already about, does that become the comparator then? If you do get to a place where you have an approved vaccine, presumably only in 2021 with the full independent scientific review, does that need to then become the comparator to the other vaccines that are currently in development but not yet in efficacy trials compared to placebo?
Well, there's also a lot of debate around that, as you probably know. And so, the mRNA came out of the blocks really fast because it's a synthetic process that can really go straight into manufacturing really fast. And I like it, or I wouldn't have chosen it, but and I like it because it's not only immunogenic, but it creates T-cell responses that I think can mitigate any kind of concern about enhancement of illness type of problems. And so I like this. It may or may not be as immunogenic for antibody as a protein. It may or may not be as immunogenic for CD8 T cells as an adenovirus vector. And so each of these new programs is going to bring a different element. And, the subunit vaccines may not have CD8s and they may not have as much biased Th1, but they're going to have very high antibody levels. And so if you get the conformation right, you could have a very potent vaccine. And the question then is how much potency do you need to protect? And, I'm counting on the fact that what we've generated with RNA, which is in the upper tier of convalescent responses, that should be enough to protect. But if it's not, then we very shortly have subunit proteins coming along that have even higher antibody responses. And we have ad vectors coming along, if CD8s are critically important, that will also have their own impact. And so if one of these works and has really good data first, it becomes difficult to know what to do, because the other problem is supply. And none of these companies can make enough to supply the whole world. None of these companies can immediately make enough to supply the United States, let alone the whole world. And so I think we're probably going to need multiple companies involved, maybe with multiple approaches to vaccination, to really get on top of this to create enough world immunity to have this thing go away or become seasonal.
Yeah. It's super important. I'll make a shameless plug. I'll just hold it up here, and it's online at AVAC. It's a cheat sheet about the pipeline that looks at these different products, and really want to encourage people to download that, and hopefully we'll find it helpful. I do want to turn running to two hot topics that have come up in a number of the questions and comments, and have been swirling, I think, for a lot of us over the last couple of months. One, and again, the work that the VRC has done, and that AVAC and many other organizations represented on the line have done, focused in HIV, and yet when the Moderna phase 3 trial was beginning, people living with HIV were excluded from the trial, and a lot of remarkable advocacy and some of the leaders of that movement on the line here. I wonder if you having worked so much in this field broadly in infectious disease and particularly in HIV, the rationale for having excluded HIV positive individuals to begin with, and just if you could clarify if that now has changed for the Moderna phase 3, and if someone living with HIV is able now to enroll in the trial.
Right. Well, I've been involved in HIV vaccines since 1987. And so you know how I feel about this, Mitchell. And, this whole process, this has been nonstop since the 11th of January. This whole process has been unprecedented in how to set up a phase 3 study, and how to set up new networks, and how to set up a new infrastructure for managing this and across government agencies. And so it's really been amazing how quickly this has happened. But there's also a lot of complex things that happen when you combine an academic network or a government system with private industry who has their own way of working with CROs and you know, basically different sets of priorities and concerns. And so some things get missed along the way, or some things get slipped into an amendment. You know, it's kind of like legislation that you may not have even noticed until later. And so it did start out that way, but it got corrected very quickly. So people who are HIV positive and controlled are eligible for the Moderna trial. I just want everybody to know that that is the case.
And really,
And so it is not an issue, and really it was just an oversight that ever got in to begin with, and probably a misunderstanding and a, you know, hyper cautious position by the CROs who were helping Moderna write this protocol. So it did slip through, and now it's corrected.
Okay, that's super helpful. I know there's someone on the line who's been tracking this closely, and I think how these decisions go from a protocol amendment to get the FDA approval. I know at least some trial sites have said they don't yet have the clearance to enroll someone who is HIV infected but controlled, so I think you know, it is a process. It's taken longer, I think, than some would like, but really helpful to know. And I guess it does raise an issue. You know, to be blunt, who's in charge? Obviously, you, Barney, and you, the VRC, have spent an enormous amount of time and energy focused on this vaccine candidate, and yet it sits with Moderna. They've obviously made some investments at risk, as you say, financially, to manufacture. But, you know, obviously, all of these endeavors require multiple parties, but ultimately, presumably it's Moderna's vaccine, or does the NIH have a role, having really, as you presented it, as a co-developer?
Right, well, it has to be Moderna's vaccine. They have to hold the IND. They have to be the sponsor, because if they aren't, they will not be able to invest in the supply and scale up and manufacturing needed to actually distribute the product. And so this is, like I say, it's a very much of a unique one, first ever hybrid between private CRO type studies and government consortiums and academic sites that bring a different level of different priorities, different skill sets, different philosophies, and, you know, to the table. And so it has to be Moderna's vaccine, but we have to, if we're investing this much government money, have to have some level of harmonization, oversight, involvement, influence, and, you know, outcomes as a government and over this process. And so, you know, there's obviously, people could find a lot to criticize here, but it's also remarkable how fast this new thing has come together. And even though there's been a lot of things to solve, it's actually happening, and we're starting to, you know, generate data that's going to be relatively harmonized, assays that are going to be relatively, you know, consistent across all the different studies, and endpoints and outcomes and decision points that are all going to be transparent and open so that we know exactly what's going on.
Yeah, Brian, I really appreciate you saying it. You know, it's interesting. There's always a lot to criticize everywhere. But, you know, and I think about all of our work for decades in HIV vaccines, and we've long said we wanted industry more involved. And when you look at that slide of all the vaccines with U.S. government financial support, but with industry clearly deeply engaged, we fantasized about that. We still fantasize about it in HIV. Right. We had Merck's great involvement early on in the Ad5. Now, of course, we have Janssen's terrific involvement with the Ad26 HIV vaccine and their COVID vaccine. But if you put up a slide, it's basically, we have one company involved in HIV and dozens in terms of really investing. I
think this is another thing we learned from HIV, is that just having a good idea or a good product concept coming out of academia does not get a vaccine into people's arms.
Yeah.
And so it's critically important to have industry involved. And to do that, we have to combine the ways of thinking and the rules of engagement with the way that, you know, government academia is used to engaging, and try to make that work. And so it is working, even, you know, even though there's been a few bumps along the way. And so it may actually help us in the future with other problems if we can really figure out the rules of engagement here.
Yeah. No, I think that's super helpful. I mean, we talked early on in a document we put out in May about what HIV vaccine platforms have done, product platforms, the VRC's own work that have helped accelerate COVID. And I think your last point here, how do we bring some of the COVID lessons back into HIV is important. I want to pick up on two things though about both planning for success and planning for failure. mRNA has not been developed for another vaccine. We have 2 in phase 3 right now for COVID. Prospects of an mRNA-based vaccine for HIV? Possible, excitable, not?
Well, I think there's a number of technologies that have been used in now RSV and influenza and coronavirus and other things, even flaviviruses, that can all feed back into maybe inform HIV. And so I think mRNA delivery is certainly a reasonable thing to use for HIV vaccines. It can induce antibodies and it can induce CD8 T cells, both of which will probably be needed for an effective vaccine. I think the real problem in HIV still lies in the antigen design and the difficulty of penetrating the glycan shield and accommodating the great diversity and, you know, in the antigens and genomes. And it is, I think it's still largely a problem of antigen design, not antigen delivery.
Great point. And I believe Moderna actually has an mRNA based HIV vaccine candidate as well, correct?
Moderna has been working with us and with and Peter Kwong, especially at the VRC, on some approaches, and with other groups as well. I think they've got multiple HIV projects with other groups.
Great. So let me ask so that we'd hope mRNA will be licensed for COVID and perhaps other things. One of the things though that you all at the VRC have taught us in HIV is to not have your eggs in one basket. VRC really led a different adeno-5 based vaccine as a lead candidate. It didn't work, and while that was disappointing, you all were simultaneously working on the various neutralizing antibodies. And we now, as I said at the beginning of the call, are waiting for the results from the 2 AMP studies of the VRC01 antibody. So from a COVID perspective, mRNA is in a phase 3. We hope for success. Do you have a plan B at the VRC? Is there a VRC program in case mRNA doesn't work? Or if it does, as you say, the need for multiple approaches, multiple products?
Yes, well, you know, our antigen design, the 2P stabilizing mutation, is being used in a lot of the platforms going forward. And but the VRC is also looking into at least 3 different ways of using protein-based vaccines, either alone or as on a particle, or different adjuvant approaches, delivery approaches that could be a backup plan. And there's a couple of other projects looking at VLPs and things as backup strategies. We put a lot of effort into this initial launch of the mRNA, but we all think that there's probably a need for different types of vaccines, and it may be that this first year that nucleic acid and vectors would be a great thing to get people primed with and maybe protected. And 3 years from now, it may be better to boost them all with proteins. And 10 years from now, it may be better to have live attenuated viruses for our children to try to get them immunized, earlier in life. And so I think as we go through this, there's going to be a need for thinking about different vaccine approaches for different types of problems.
Yeah.
You know, the boosting problem, the children problem. We need vaccines eventually that can work and easily in pregnant women and different special populations that have immunocompromised states. And so there's a lot of different types of things we need vaccines for. And at the same time, we're also working on monoclonal antibodies, and they may be able to fill some of those gaps, but I think there's still a lot of things to figure out here.
Yeah. Sounds remarkably like HIV, Barney.
Except that we can make neutralizing antibodies.
Yeah, exactly. Exactly. The science seems a little bit easier, but many of the same themes at least. One of the other hot button issues that I mentioned earlier, I was going to ask you too. But one, and I want to, I know we have people dropping off, and we're going to come to an end in just a second. But all of the trials you described in people, not the mice and monkey trials, but all the human trials, and all the phase 3 trials, including the one you all are involved with, the Moderna product, are all classic randomized control trials, placebo-controlled trials. And I want to make that really clear, if it wasn't some people on the call, because there has been a lot of conversation, a lot of articles written about the idea of a human challenge study, and the idea that somehow that could speed things up. And a number of us, in full disclosure, AVAC, working with the Treatment Action Group, and a number of other advocates have been very clear, not to oppose human challenge trials full stop, but to be careful with our language. There, at least to us, it doesn't seem there's absolute clarity that it would get a vaccine sooner necessarily, and particularly without a challenge model validated for humans, and without a good treatment for people. But I wonder if you could just describe maybe your thinking about human challenge trials, the role that they might play, if any, and how you and the VRC might be thinking about a potential role, if at all.
Right. Well, for the sake of humanity, I think people should remember the show on NPR called All Things Considered. There's so much coronavirus disease right now in the world that it seems improbable that we would need or use a challenge study, to me. And so we don't have a drug that can easily treat coronavirus disease. If we had an antiviral, that would make it a lot easier to consider that. If coronavirus disease disappeared and we were afraid it would come back next winter, and we couldn't get an efficacy answer ahead of time, that would make it easier to consider. All these things are very things that we should consider. But, with this much coronavirus disease, it seems like we should easily be able to get an answer in the field. And but in the meantime, I don't think there's anything wrong with talking about it or discussing it. If we had to use it, what would it mean and what would it look like? Or even taking the trouble and the expense and the time to make a challenge stock by GLP methods and GMP stock that could be used, even though we would never use it. And so I think all those things in the most conservative sense should be maybe done and considered, but I don't think we'll need to use it. And the VRC is not planning to use that. I don't think we'll need it, and hopefully we'll be able to identify a correlative immunity that would allow you to then even not have to do such big field studies and maybe just bridge to a correlative immunity. And so I don't think they will be needed, but I think we should discuss them and prepare for them just in case.
That's super helpful, Barney. And I just put in the chat for attendees some guiding principles that TAG and AVAC put together. Obviously, we come at it from as non-scientists, but I think, Barney, we landed in exactly the place you did. Not to be taking anything off the table, but let's not assume that that's a panacea or needs to be done right now, and a number of issues that you outlined. So it's always
but
nice
also not be afraid to talk about it.
No, absolutely, absolutely, and I think that's exactly whether we go deep into the science or deep into the advocacy issues, there's clear common ground, and I think figuring out how we do this with utmost speed without cutting corners, without sacrificing, as you said early on, that you all have moved with unbelievable speed, but without compromising scientific rigor. And I think that's something that can get lost. I think people get nervous when they hear things moving so quickly. And I really appreciate your framing the work and making sure that no one step happened before you had data to inform that decision point. I think that was beautifully described. I know we have left a lot of questions unanswered and even unasked, and I apologize. Obviously, lots to talk about. Delighted to have almost 100 people still on, 30 minutes overtime. Barney, just any last thoughts, perspectives from you before we close out?
Well, what I always like to remind people of is the reason this could have worked so quickly is because of prior investment in basic research. And investing in basic research and understanding the biology of these viruses and doing that ahead of time, somewhat at risk because maybe these things would never become a problem, but I think it's so important because, you know, they speak across platforms. RSV informed, well, HIV informed RSV, RSV informed coronaviruses, and now they're both informing back to HIV. And so I think this concept of investing in basic research and biology almost always has a fruitful outcome. And I think it's very hard for people to make decisions on investing in basic research and preparedness, you know, before they see the need for that thing to happen. And I just think that is a big message out of this that I think people should remember.
Yeah, Barney, it's a brilliant message, and I'm so glad you said it because it's often harder, I think, for advocates and policymakers sometimes to wrap heads around that basic science. But I think you've really demonstrated it beautifully, and the VRC has done in 23 years now of what it means to make that investment. And I think there's a lot of work for all of us to be doing going forward, that we don't wait for the next the next pandemic, because we will deal with coronavirus, we will deal with HIV, there will be something else. And I hope collectively that we figure out the infrastructure and the investments that need to transcend disease or virus categories. And I think you really presented here.
So a couple of billion dollars a year for the next 20 years could put us in a fundamentally different position for any other threat.
Yep.
And that amount of money seemed overwhelming 6 months ago.
Yep.
And now it sounds pretty trivial.
Yep.
And I just think that we just need to go ahead and do it.
Yep. As usual, Barney, you have made the advocacy pitch better than the advocate. I could not agree more, and really look forward to continuing to collaborate with all of you to monitor and watch the science unfold over the next couple of months, and more importantly, as you rightly said, for the next several years and decades, because we've got a lot of work to do together. I can't thank you enough, and you will, and the entire group behind you in the VRC. Clearly, as you well described, it takes a lot of people moving this forward, both within the VRC with all of your collaborators. We're delighted to see the progress and to have a chance to talk with you. And this is not the first time you've helped AVAC and colleagues understand, and it certainly won't be the last. So we really look forward to that. So thank you. Keep it up, and we will as well. I want to thank all of you for sticking on the call. Incredibly helpful to see your questions. Again, apologies that we didn't get to all of them, but do keep in touch. We'll continue to answer them. This will be the first in a series of webinars with other of the vaccines in development as all of this unfolds. And again, some materials just posted yesterday at the AVAC website to help understand the pipeline as it exists and to understand the risks and benefits of how we do this better, faster together. So thank you all for joining. Barney, again, thanks so much. Stay safe and well, and we will talk very soon.
All right. See you all.
All the very best.